speaker is Michaela Leung. Michaela is a fourth-year PhD candidate in the Department of Earth and Planetary Sciences at the University of California, Riverside. Her main research interests are atmospheric biosignatures, terrestrial atmosphere photochemistry, and the intersections between Earth and exoplanetary science. Her other professional interests include developing inclusive communities and classroom pedagogy. The title of her talk is Methylated Biosignatures, a New Class of Biosignatures. Michaela, whenever you're ready, go ahead and take it away.
Alrighty. Thank you so much, Tiffany. So like Tiffany said, my name is Michaela Leung, and I will be talking to you today about my work on specifically thinking about new gases that we can use as potential biosignatures. And so this was kind of set up a little bit in the last talk, but one exciting thing that we can do with all these exoplanets that are being discovered now is kind of this search for another planet like the Earth. And for many people, the kind of ultimate goal of such a search is to find another planet that has life on it. And so what you're looking at here is a simulated spectrum of an Earth-like exoplanet where we do see these potential biomarkers like methane, oxygen, and even like carbon dioxide and water kind of all shown in the spectrum. And a lot of what you're seeing here in this concept art are really kind of the most well-developed potential biosignatures. However, as we are kind of waiting for new instruments like HWO and even, various observations to come back, there's still a lot of other possible biosignatures that should be explored so we can kind of maximize our chances of being able to identify other life if it's out there. So that's kind of the vein in which I'll be sharing more about my work today. Just as a quick table of contents, I'm going to do some background and motivation. And then there's kind of two main parts to the results for the talk. I'll be talking about halomethanes as biosignatures, and I'll also be talking about methylmercury as kind of a sample of a larger group. So you might be wondering, the title of my talk has to do with methylated biosignatures, and you might be kind of like, well, what does that even mean? And so, a methylated biosignature is formed by the addition of a methyl group, which is a carbon and three hydrogens, to kind of some sort of additional compound, often just kind of an atom that's been sitting around in the environment and is most commonly done as a biological process to remove those other elements kind of from the living conditions of a microorganism. And so then you get this methylated molecule. And you can see in this lovely art we have here on the slide that these gases are often produced in both marine and terrestrial ecosystems by microbes and even some plants. And they are then fluxed into the atmosphere where they interact with other chemical species, as well as the light from the star. And you can see in the corner here, hopefully you can see that, that the type of the star can be really important for determining what happens in the atmosphere photochemically. And then we also consider how these gases impact the overall spectrum of the planet with a specific eye towards, how could we potentially detect these gases on a planet if they were there. So jumping into a little bit of the motivation for pursuing this specific group of gases. So firstly, we think these gases have really low false positive potential. One of the things about some of the more traditional biosignatures that I was highlighting earlier, things like oxygen and methane, is that they can be produced in a number of ways by abiotic processes, which does make them a little bit more difficult to interpret as biosignatures were they to be detected. However, these methylated gases are not produced as equilibrium products in high temperature, high pressure atmospheres, which is one way you can produce some potential biosignatures. And you also get pretty minimal quantities from planetary processes. And this is things like volcanism or water rock reactions that on the Earth can produce, again, some of these potential biosignature candidates. And like many other gases, you can produce methylated gases from disequilibrium dust grain chemistry on the surface of comets. And so if you flux the surface of your planet with, massive amounts of comets, you could potentially get a false positive signal. But this is most likely to occur in a really young or dynamically unstable system, which is kind of an unlikely target for a biosignature search anyway. So as a kind of a combination of each of these factors, we really think the false positive potential for the methylated gases is pretty low. We also have found that this process is very widespread. So we do see methylated gas production, most productively actually in these sort of marine terrestrial crossover ecosystems like wetlands, salt marshes, kind of bogs. But we do also see production in the open ocean, on glaciers and microbial mats and sewage in a pretty broad variety of different environments here on the Earth. And one of the reasons why that is because it's actually a process that is done by a pretty broad variety of organisms. Like I say organisms, it's primarily microbes, but it is a number of different groups of microbes, which suggests that it's, a common and or useful process. And one fun little plug is that the brassica group, which contains like everyone's favorite vegetable, does also produce methylated gases. So that's a little fun tie-in. And there's a little bit of previous work on methylated biosignatures as well. The first part of which, from 2005, does show that you get enhanced atmospheric accumulation around M dwarf stars, which are popular targets right now, especially using JWST. And this is because these M dwarf stars have lower NUV flux, which ultimately decreases the sinks for methylated gases. And some other previous work has also shown that you can get a secondary biosignature for specifically dimethyl sulfide and dimethyl disulfide. And this is basically showing that even if these gases are not directly detectable, in some cases they may contribute to kind of, like I said, a secondary signature that potentially could be picked up as well. So all of these factors have kind of motivated us to pursue this methylated gas group a little bit more. And so I'll be talking now about our evaluation of halomethanes as biosignatures. And so halomethanes are just members of this methylated halogens group, which you can see highlighted in the little orange box on the left side of the slide. But we're looking at gases in the halogen group, which is specifically chlorine, bromine, and iodine. And the halomethanes that I'll be talking about today are just sort of like the methyl chloride, methyl bromide, and methyl iodine. And we do hope to explore some of these other methylated gases, and I'll talk a little bit about that at the end of this section. So to understand these gases as potential biosignatures, we're using a combination of modeling methods. And so the first step of that is to do photochemical modeling to understand the atmospheric composition. And then the second part is to use transmission, emission, and instrumental modeling to understand basically how these gases could potentially impact detectable parts of the atmosphere. So I'm going to jump right in with some of our photochemical results. So these were published in a paper a few years ago, and we're looking here at a variety of fluxes and stellar types. And kind of just doing a kind of a sweep in FGK M stars for, like I said, a variety of flux conditions. And what we're showing here is we do replicate the earlier result, suggesting that M dwarfs do produce more, or don't produce, but they allow for a higher accumulation of methylated gases in the atmosphere, and particularly with methyl bromide, which had not previously been explored with these photochemical simulations. We do see an even more significant atmospheric buildup, which is really exciting because methyl bromide does have a little bit more of a modest flux here on the Earth. So it's really exciting to see that even larger potential buildup. And like I said, we are exploring a pretty broad range of fluxes here, but actually these gases are so spatially variable on the Earth that in some of these kind of local environments, you do actually see up to 1,000 or even a few thousand times the globally extrapolated flux. So we do think that that actually is a pretty reasonable assumption for the constraints of biology as we sort of see it here on the Earth, and just kind of imagining a world where you perhaps have more of these highly productive ecosystems. And so building off of those photochemical results where we see the buildup for M dwarf stars, we also are looking here at some mid-infrared emission spectra. And on your left side, we're seeing a set of kind of planetary spectra. And next to that, we're seeing the input spectral features. And what I really want to point out here is, and you can see this especially well in the kind of input side, is that the methyl chloride and methyl bromide features do kind of pretty neatly overlap. So what this contributes to. You can kind of see this in the bottom row of the planetary spectra, but this contributes to creating a much broader combined methylated gas feature. And this is actually more on the photochemical side, but when you have multiple methylated gases present, we do tend to see a higher buildup of each of the gases, just because they have the same, sinks, the same destructive processes. And so when you have multiple methylated gases, you can get higher buildup of each individual gas, and then these larger, potentially more detectable features overall. And so what I just shared was the methyl chloride and methyl bromide results, and this is actually a new preliminary result for methyl iodine. And right now we're just considering the methyl iodine. But we hope to also explore these kind of other covariant levels of both the other methyl halides that I talked about a minute ago, as well as some of what are called polyhalomethanes, where you have multiple halogens that have been attached to this kind of similarly constructed molecule. For methyl iodine, we are seeing the same overall trend with accumulation towards the M dwarfs, although the buildup here is a little bit more modest than what we were seeing before. Like I said, this is a preliminary result, but I kind of think at this point that it's because this is a larger molecule that is, it's heavier, it's not maybe rising as high in the atmosphere, and it's being destroyed by more chemical and photochemical processes, just because it's bigger, and that does relate to the amount of photochemical destruction that occurs. And we do hope that the methyl iodine will also contribute to this co-additive feature, kind of similarly to what I was showing before, with the methyl iodide now in green. It's another kind of overlapping feature that we hope will contribute. And here I showed before an emission spectrum that we had simulated with methyl chloride and methyl bromide. This here is a transmission spectrum for something like we originally simulated for the origins concept, but it could be useful for any sort of mid-infrared capable telescope. And you could see again between 10 and 12 microns. We do have the methyl chloride, the methyl bromide, and again, that larger combined feature. And I did some preliminary simulations for methyl iodine at about a 1 part per million level. And what you could see on the right here is this is again in that 10 to 11 micron window range. And around 1 ppm, we do start to get this kind of bump that might be potentially detectable or contributing to the overall signal. And likewise, between 6.5 and 7.5 microns, we are seeing a little bit of a bump there as well. So we're kind of hoping that these pieces will add to those overall combined features and again enhance kind of those pieces as well. And we hope also that the sort of more when we include more gases in the simulation, that we may also see a higher buildup of a methyl iodine that will allow it to contribute more, since we have seen that trend for methyl chloride and methyl bromide. So I showed that this spectrum that we had originally simulated for the Origins mission, and we also have done some simulations in collaboration with the LIFE mission team. The LIFE mission is the Large Interferometer for Exoplanets. If you're not familiar with the mission, it uses a set of smaller telescopes flying in formation, and then uses interferometry to kind of enhance the signal from those telescopes and contribute to some better detections. And so we did some simulations for a TRAPPIST-like system. TRAPPIST is, of course, every biosignature enthusiast's favorite because there are a number of favorable advantages because the star is so small. And so here we simulate actually a TRAPPIST-like planet at 5 parsecs, so a little bit more optimistic than TRAPPIST itself. But kind of the main takeaway here is we're exploring, again, a range of those methylated gases. I should say here that this was done with just the methyl chloride and methyl bromide, so that's what we're looking at here. And this simulation is for 10 days of observation, and you can see that even at just 10 days of observation, we can detect that combined feature around 10 microns at about 3 sigma confidence, which is really great. And we also did some other simulations for like longer periods of time, and this is for 100 days. And you can see that we have even more significant confidence in the size of these features, and that we could get a really strong observation of these gases potentially with a longer observational duration. If you're interested in hearing more about this, we do have a paper out that it's in AJ, or you can scan the QR code, but it's a really cool concept, and it's probably one of the best ways to look for these gases in the mid-infrared. So definitely check that out if you're interested in hearing more. We do also have some future work. So in addition to the methyl iodine that I'm currently working on, we also are interested in kind of these super Earth planets, particularly because with JWST, we are kind of right at super Earths as kind of, I think, the limits of these more favorable targets. And there's a lot of other work going on right now looking at biosignatures on super Earths. And so we're hoping to also explore the methylated gases in these super Earths. I do have a very preliminary kind of sneak preview result which was actually generated using the Vulcan photochemical model, and specifically using a Shami size 2024 paper K218b template. And what you're looking at here is just the atmospheric mixing ratios of all of the gases. And this pink line here, which is the methyl chloride, is actually showing a surface mixing ratio of about 40 parts per million, which is about 2 orders of magnitude higher than what we see on an Earth-like planet. And so what this is just for like the globally averaged Earth-like flux. So this does suggest that these targets might be extremely favorable for potentially observing methylated gases kind of in the near future, doing transmission spectroscopy with JWST. So that's really exciting, and hopefully we'll have the rest of the gases evaluated and out soon. So definitely keep an eye out for that. So kind of the takeaways from this section, methylated halogens can be really advantageous as potential biosignatures, and specifically this combined methyl chloride and methyl bromide feature might be detectable with future mid-infrared missions, such as the LIFE concept, which we explored here, and that we're also hoping to explore methyl iodine and polyhalomethanes, as well as looking at super Earth atmospheres as kind of an alternative environment in which you might look for biosignatures. So that's kind of the pieces of this section. But like I kind of mentioned earlier, methyl halides are really only a small part of this group of methylated gases. And I'm actually going to transition now to talking about something from this red section over here, which is actually methyl mercury as a sample methylated metal biosignature. And we are using, again, here the same sort of overall structure, but the first step here is actually to implement flux measurements that were taken in the laboratory for a variety of atmospheric conditions and use those to inform the rest of the modeling pipeline. And so I'm just going to show some plots that are very similar to what we were just looking at for the other gases. Nope. Our flux measurements, though, were contributed by our collaborators Emma Yang at Oakland University, who works on Arctic soils and methyl mercury measurements. And we looked at a variety of atmospheric conditions and 2 temperatures. The 4 degrees Celsius is shown in like kind of a pale blue color. And one of the main things you'll notice here is that the methyl mercury production is way higher at like 30 degrees Celsius, which does mean that for a sort of more temperate exoplanet, you might get these much higher fluxes versus what's been recorded in the environment on the Earth. And also, we do see that our most productive cases incorporate either like 100% nitrogen or have high levels of hydrogen, which we were just kind of talking about these super Earth planets. And those are 2 kind of popular suggestions for what those planets might be like. So the higher fluxes of methyl mercury for those planets might be plausible in super Earth planets. Okay, now I'm going to talk about the photochemical results. So actually, for the photochemical results, the first thing we did was look in the literature and see kind of, what's the globally average flux for methyl mercury here on the Earth? And the answer is not a lot. For the values that we were able to find in the literature, we do get kind of a most optimistic buildup of around 1 part per trillion, which is not necessarily a level that we would expect to be detectable in an atmosphere. However, when we incorporate the fluxes from the laboratory measurements that Ziming's group conducted, we do see a significant increase where we're getting close to 1 part per trillion for just the sort of base Earth around the sun case, and that we are getting up to tens of parts per billion for, again, our most optimistic TRAPPIST-1e and Proxima Centauri b cases. And I'll just flip back and forth, right? It's a really significant kind of jump that we see in the buildup, which really kind of just goes back to these input fluxes that we have. And so kind of our next step is to look at these Hycean sort of hydrogen dominated atmospheres in a more self-consistent manner. So we've actually treated the hydrogen gas biological flux as kind of a biological limit. We've still explored these in an Earth-like atmosphere. So we also hope to do the self-consistent Hycean simulations where we also are changing the bulk composition of the atmosphere. And based on the result that I showed for methyl chloride in the Hycean world like a few minutes ago, I do expect that this could potentially also significantly increase the overall fluxes of the overall mixing ratios that build up. And then we also potentially may perform spectral simulations if that increase does rise to a more significant level. And we do have features ranging all the way from kind of like 2.5 microns up to 22. And so at the short end, these would maybe be accessible to something like JWST for maybe a super Earth. And then at the longer wavelengths, this would be another good fit for the life mission that I was talking about earlier. And this sort of like integrated work combining the fluxes and the modeling, I think, is really important. And we're actually pursuing doing some other field studies at the Salton Sea, which is an analog site in Southern California. And we're hoping to collect more gas flux input data that we can use to, again, kind of better inform and enhance both our understanding of kind of how much these gases can build up, and also to think a little bit more about the environments in which these gases are produced, to kind of better understand the applicability of the gases as biosignatures. So my kind of takeaways from this section is that these laboratory and field measurements are really useful to explore, again, these alternative planetary conditions, which are critical for understanding the biosignature application, the degree to which they're going to actually be detectable, and kind of basically their overall usefulness. A specific result that we do find is that methylmercury production is greatest under hydrogen-dominated temperate conditions. While our results so far suggest that it's right kind of on the cusp of maybe being close to detectable, we do think that, if you did have high production of these gases, because a lot of these methylated gases are toxic for more complex life, that even if you were not able to detect these gases, this result does suggest that if you did have high fluxes of methylmercury, that could potentially inhibit the formation or survival of complex life due to the toxicity of the compound, and that we have, again, some further work planned to explore self-consistent Hycean simulations for the methylmercury. So I have some acknowledgements. My advisor, Eddie Schwederman, our collaborators, Nikki Thomas, Jami Zaming, Michaela, and the rest of the Schwederman lab here at UCR. And I will leave up my conclusions for you. I have my papers on the life mission and on methyl bromide linked. And my final plug that I will leave you with is that I am graduating next June, and I'm looking for a postdoc to start summer to fall 2025. So if you're interested in this work, please reach out to me. My email is on the slide, and that QR code is for my website. And with that, I'll be happy to take any questions. Thank you.
All right. Thanks so much, Michaela. And I apologize for mispronouncing your last name. So I appreciate you correcting me. All right. Any questions? Again, you can put them in the chat or use the raise hand tool. I already see one. Neil Turner. Go ahead, Neil.
Thank you for a very interesting talk. So we're venturing far beyond what I know, but just wondering with the lab experiments, so the source of those of the metal and carbon molecules, if it's a terrestrial microorganism, might the novel atmosphere kind of produce some sort of transient response from the microorganisms? Could you say a little more about what's involved in those experiments, basically?
Yeah, so for the experiments that I mentioned earlier, they're taking basically samples of Arctic soil and putting them in like closed top bottles, is my understanding, and then incubating them for a period of time and then measuring the fluxes that are produced, specifically in a time-dependent manner, because that's really important for the photochemistry. I'm not sure if that answers kind of the first part of your question. I think we do expect that like if the gases are short enough lived in the atmosphere, right, that you would get a more transient signal. But I think the idea is if you have these gases coming from like a biologic source, they're producing sort of like round the clock, so to speak. And so you would maybe get a more consistent signal. Does that answer your question?
Partly, I think. So I'm wondering if, like, what's the reason for the microorganisms to emit this gas? I mean, you alluded early in your talk to getting rid of toxins or other things that they don't want. So I'm just wondering whether you expose them to this non-terrestrial atmosphere, maybe that changes what they want to get rid of. So I'm wondering if they were in that atmosphere, if they had evolved in that atmosphere, perhaps they would have different needs and produce gases in different amounts.
Okay, yeah. I see what you're saying. Yes, I think that is, obviously we are not able to evolve the microbes under unique conditions, unfortunately. That would be really cool. If we were able to do some sort of, really long study that looked at how, a completely different atmosphere would shape the overall evolution of these microbes. What I will say is there is some evidence to suggest that the methylation is used as a detoxification process, which is possible, possibly in need of life, just to kind of, control its environment, and to be able to continue to grow by removing some of these metals and things like that. The other thing I will say is that the methylation process in some cases does come uses the same enzymes as DNA methylation, which is actually on Earth a pretty ancient process that we think dates back possibly as far as the last universal common ancestor. So we kind of think that this process is, very old and possibly again related to detoxification under certain conditions. And so it's kind of, I think, between those two factors, we're sort of hoping that makes it likely to occur on an exoplanet. But yeah, I mean, we obviously cannot say with 100% confidence what would happen under like fundamentally different biochemical conditions.
I see. Thank you for explaining the challenges and the reasons there. Thank you.
Thanks, Neil. And it looks like Nikki added some additional thoughts on that front. So you can take a look at her chat message. All right. Ilaria, go ahead and ask a question.
Hi, thank you very much for your talk. I was interested in the simulated spectrum for the methylated biosignatures. So I was wondering about the water vapor, right? So there should be also water vapor there. So did you include it, and what is the effect of increasing or decreasing the water vapor to your signature?
Yeah, that's a great question. We haven't specifically looked at changing the water vapor. We do have water in the input gases that kind of go into the spectral model, but we have not looked at the effects of changing that profile. So it is included, but we're not changing it so far. So that's definitely something to look at in the future.
Okay, thanks.
All right. Neil, you still had your hand raised. Did you have another question? Okay.
That was a thoughtful.
Yeah, no worries. All right. Any last questions? I guess I'll just ask a quick one. And maybe I know I suspect I know the answer, but I just wanted to ask. I mean, you mentioned some of these methylated signatures, would be detectable with the life concept. For some of these, it sounded like you were going to explore these methylated signatures for Hycean worlds, and I know those are getting a lot of attention with JWST. Is there any expectation that you could detect some of those features in certain planets with JWST, or is it really like photon starved, or what's the limiting factor, I guess?
So we did originally in the methyl bromide study do some simulations using JWST parameters, and the problem is that we're in the mid-infrared, and my understanding is that the noise on MIRI, this was actually before the telescope went up, so maybe we know more that changes that now, but in all of the simulations that I did, it was like 1,000 transits to detect. So obviously you can do it, you just gotta like
invest a ton of time.
Obviously that's not a realistic idea about what's going to happen with the telescope time. Perhaps a little too far. And people will often say like, oh, 100 transits, 1,000 transits is a little too far. That's longer than the nominal lifetime of the telescope.
Yeah, sure, of course. Okay, that was my suspicion. I just wanted to ask a question. But yeah, I mean, I can't imagine that the our new understanding of the noise floor and so on would really change that answer significantly, I would think.
Yeah, that's my understanding as well from kind of what I see go by.
Got it. All right, thanks. All right, any last questions? I don't see any other hands raised.